I have the great pleasure this year of helping the society find presenters. And so it was with great excitement that I found Alina. And is that the pronunciation, Alina?
Yes.
Yeah, perfect. Thank you. And totally coincidentally, like the day after, the next day in the Washington Post, in the weekend section, I learned something that I didn't know. And I've been in DC for 40 years, that DC has a DC Chocolate Society. And I wasn't going to mention it if they met on Tuesday nights and that it would conflict, but I just thought if you didn't know, there is a DC Chocolate Society. And I don't know, many of you know, I was at the U.S. Botanic Garden for many years, and I always used to talk about our Theobroma cacao as the panda of the plant world because people could relate to it so well. And so I am going to let Alina introduce herself and talk about her background. But we are thrilled to have you with us this evening. And this is a great group of people, really informal. And so you can decide if you'd like to take questions as we go along. I'll monitor the chat, or if you'd like to reserve that till the end and, just see how you feel. Yeah, so thank you so much for being with us this evening.
Okay, yeah, thanks for inviting me. I'm glad it worked out. And yeah, it's nice to meet everybody. I'm currently in my office at Fort Detrick. I think I'm the only person in the building, but let me go ahead and share some slides with you. Oh, so to introduce myself, my name is Alina Puig. I'm a research plant pathologist at USDA ARS. I've been with USDA ARS for about 9 years. Primarily, I've worked on cacao for about 5 years, and now I work on emerging viruses and oomycetes of a range of different crops, including cacao, mango, avocado, also potato and corn. So a big range of diseases. So let me go ahead and share my slides. Let's see. Can you see the presentation?
Yeah, I think you need to make it full screen still, but
Oh, is it not full screen?
I don't know. I can't tell. Oops.
Let me see. Hang on. Let's see. I went through this before I started to make sure I knew how to do it. So let's switch
where you were before. All you need to do is hit the full screen thing on your PowerPoint, and it should.
Got it. There. Can you see that?
Nope.
Not yet.
Can't
Nope.
see it. Okay, let's see.
Go back to share.
Let me share this again. Okay, give it a minute, see if it goes into full screen mode. Can you see that?
It's not full screen, but
it's not full screen.
It might be presenter mode. We might be seeing the presenter mode.
Oh,
Yeah,
you're
we are
seeing
seeing
that.
presenter mode.
Okay, let me switch them out then. Let's see, display settings. How about that?
That's
perfect.
perfect. Wonderful.
Okay.
And if I
have
could
done
just
this
remind
before.
everybody to mute, that would be great.
Okay, perfect. So I'm going to talk today about Theobroma cacao cultivation and diseases. My specialty is I'm a plant pathologist, so I work primarily with the diseases. And Theobroma cacao, as you all know, it's a tropical tree. It's a tropical tree. It has I'm not sure how many of you have actually seen it in person, but the fruit they're like pods. They almost look like pumpkins. They grow directly off the tree itself from the stem. And when you cut it open, depends on the kind of cacao, but it's usually fairly thick. In the center here, you can see what the open pod looks like, the seeds that are what you use to make chocolate, which is what it's best known for. And that white pulp on the outside is a sweet pulp. So in some places, particularly in northern Brazil, it's used for juices. Other places it's dried and used in candies. But those sugars are what fuel the fermentation process. That's what gives cacao the flavor. And on the right side, you can see the flowers, how they grow directly off the trunk, and you can see some very small green pods developing. So this is what it looks like when it's in the field. You have a range of pod colors. They can be orange, red, or green. They can also be a pretty wide range of shapes. So here on the top left, these photos were taken, the whole top row are photos that I took when I went to Ghana. On the left is after the harvest. So they go and they open up the pods in the field there. Each country has they do it slightly differently, but this is how they did it in Ghana. And they would open up the pods, the pulp, the whole mass with the seeds and the pulp would be put into containers, and then those would be taken to where they would be fermented. And so in the center, this guy, he would roll in this contraption he made, the seeds with the pulp, and he would leave it in the sun. It would ferment directly in there. And he had already calculated how long it took for his seeds to ferment. It varies a bit based on the kind of cacao, because some of them have more pulp than others. So there's an art form to how long and what methods you should use to ferment cacao. Then after it was fermented, he would leave them out directly in the sun to dry. And they would eventually be roasted. And after roasting, in the bottom, you'll start getting like a little skin that comes off. That's usually what it looks like after you roast it, and you remove that, and just the inner portion, it moves along to the chocolate making process. I know a lot of people are interested in chocolate making. It's a pretty complicated process that I don't understand. I work with the plants themselves, so unfortunately I won't be able to really go into detail on how the actual chocolate is made, just the production. So often here on the left you can see the latest grouping. I believe it's now 11 groups of cacao genetic groups, and you can see the diversity and where it is on the map in South and Central America. The general consensus is that it originated in the Amazonian region. So generally, the places where you have the most diversity of the plants is going to be where it originated. And so you see the most diversity in the Amazon basin. The cacao that has been in in Central America and Mexico, they tend to mostly belong to the Criollo group. So soon, I believe it's been about 5,000 since 5,000 BC that they found evidence of it being used just from residues in pots. And so it's been used for a long time. The first evidence of actual cultivation in small farm settings would be found in Mexico. And on the right, you can see the map of the movement. And these movement maps of the crop were developed based on residues from pottery. They looked for, I believe the compound that they looked for here to develop this was theobromine, which is present in Theobroma cacao. That's why they share the name of the root of the name. And then it was moved to West Africa, and it was also moved to Asia. And it's moved around a few times. There have been some places, particularly in the Caribbean islands, where it was introduced more than once, because the first time it died out for either hurricane or diseases. And so there's within the genus Theobroma, there are now 23 accepted species within that genus. And again, mirroring the origin of Theobroma cacao, you can see that within the genus, that's where most of the species richness is found, in the same area where the Theobroma cacao originated. And at the top, I put some of the fruits just to get an idea of the relatives. So you have Theobroma bicolor, Theobroma grandiflorum, and then you have the Erraña species. So there are several of them. They're considered very, very close relatives, even though they're split into a different genus. And they also have flowers that develop directly on the stem. The only other one I know that they make any chocolate-like products out of would be the grandiflorum, which is called cupuaçu. They do use the seeds to make something that's similar to chocolate. It's called latte. I personally am not a fan. It's an odd flavor, but it's a similar texture. And here on the right are the most recently discovered species belonging to the genus Theobroma. I believe that was published a few months ago, and it was also in the Amazonian region. And there are 3 new species that were recently described. So even though this evolved in the Amazonian region and the Americas, the vast majority of global production is going to be in West Africa, particularly in Ivory Coast and Ghana. So you can see in the blocks on the left, they give you a sense of the proportion of each country. And Côte d'Ivoire is by far the biggest producer of cacao. That's where most of it is, followed by Ghana. And in Asia, it would be Indonesia is the largest producer. Ecuador and Brazil are the largest producers in the Americas, but they still, I want to say that it's gone up. And I think the most recent number I saw was about 19% of global production is in the Americas. Part of this reason is how similar to rubber, where the plant is moved out of its native range to places where there are less diseases because they didn't co-evolve with the plant. So those of you who are familiar with rubber, there are a lot of diseases present in its center of origin that really limit commercial production. And those diseases aren't present currently in Asia or Africa. So with cacao, it's a similar situation. It was moved out. It's considered, there are less, some of the major diseases that limit production aren't present in Africa and Asia. So generally people refer to cacao as a shade plant. And on the right, you'll see some of the planting. This is the photo on the left is from Puerto Rico. And many of the commercial growers in Puerto Rico, they planted in a shade system under native forest. And there have been studies that looked at how it affects, so that the way you plant it, whether it's shade or full sun, will affect other components. You get higher bird populations, and it protects some of the wildlife. But unfortunately, depending on where you are, if you're in West Africa, you also get more disease problems of a specific pathogen they have over there. And on the left here, you can see photos of full sun plantations. Right now in the Americas and Indonesia, most plantations are going to be large, pretty advanced plantations in full sun. So the plants, even though they do experience more stress over their lifetime, they have greater productivity when they're planted on full sun plantations. It often requires supplemental irrigation that isn't needed in the agroforestry systems. And the cacao trees, they produce a lot of flowers in a lot of locations. It's been noticed that only a minority of the flowers are actually producing pods. So one thing that has become common, varies on the country, is people will go out to the field and they will hand pollinate flowers on commercial farms. The natural pollinator it's an insect that looks like a mosquito. It's called a midge. There's a few species of midge that pollinate this flower. And so it'll go in and pollinate us. But in addition to that, commercial farms often employ people to hand pollinate. And so for that, if you look on the right, you can see that the forceps, where you remove part of the petal to cut the anthers, and the flower has to have opened within usually about 12 hours. If it's opened more than 24 hours, it's no longer considered viable. And you take the anthers to another flower and you attach them to the style. And then that one you should be able to see whether it took or not in about a few weeks. You'll start seeing some changing in the morphology of the flower. Most trees aren't self-compatible, so it's recommended to not pollinate flowers on the same tree, to take flowers from one tree and go somewhere else where you have different kinds of tree and plant them there. Normally on the larger plantations, people pick combinations of plants similar to avocado, where they know the plants are compatible with each other to increase the amount of fruit set that they'll get. So cacao production in the US, there actually is commercial production in the United States. It doesn't compare to obviously West Africa or South America, but within the US, Hawaii is going to be one of the main states that produces cacao. They're slightly out of the optimal geographic range for growing it. Cacao is considered a strictly tropical crop, and so it's between 20 degrees north and south of the equator. The places in Hawaii where it's grown are just outside of that range. And also they tend to be high elevation because it's mountainous. So the plants there, they endure colder conditions. So they do have the plants, they're doing field trials specifically in Hawaii to make sure that the plants that are being recommended to farmers for commercial production can withstand and actually produce well in those conditions. Because even if the plants survive the colder temperatures in the mountains, they don't always produce very well. So there are field trials. On the right was a field trial established by the USDA in conjunction with Mars. It was a very, very pretty research. I'd never seen such pretty field trials, but there were very pretty field trials there, and they were selecting them based on the performance of those clones in these environments. And there is the USDA does have a germplasm collection of cacao on the Big Island at their research station. So that is maintained there. And a lot of the producers in Hawaii, they're starting to also make their own chocolate. So they're starting to process their own cacao beans and making their own chocolate. And many of them have also opened up their operation as a tourist destination. And there is some commercial production in Florida. Before I moved up here to Maryland, I worked at USDA ARS in Miami. And south of Miami, there was a farm called Patch of Heaven. And they grew cacao from seed. So it wasn't a specific kind of cacao, but their big milestone was that they had finally harvested enough pods from their farm to where they were going to make chocolate using only Florida grown cacao. And but most of their other, this company does also make chocolates from using beans from other locations, but it was a big milestone. And they have people that go to the farm, and they let you see their operation. It's pretty small, but it is a commercial production in Florida. It's the only one I'm aware of. Puerto Rico has a history with commercial production. The first introduction to the island was 1625. It became a widespread industry. It was a pretty major source of income through the 1700s. And the historical records aren't quite in agreement for what happened that wiped the industry out. They use terminology that isn't used anymore today. So the best guess is that it was a combination of either hurricanes or widespread disease of plant disease, also combined with food shortages. So the hurricanes led to food shortages, and they think that that's why people didn't replant their cacao trees. They started planting food crops. But there is a little bit of disagreement on what the source on why the industry was wiped out, because it was pretty significant. And probably, I believe it was about 2008 to 2010, USDA, they have a research station in Puerto Rico, and they were doing field trials to see which clones could be planted commercially on the island and do well there. And they picked 10 clones to recommend to commercial farmers. And there's a company from the Dominican Republic called Cacao Cortez. And so they partnered with USDA, and they obtained a grant for the specialty crops. And they identified farmers on the island that were going to establish plantations using material from USDA. So there were new plantations established in 2010. Unfortunately, it was going very well until Hurricane Maria, and a lot of the farms, their trees were destroyed, right? And it was right when they were getting to the age where they were finally getting good harvests. So there was a big initiative to replace the trees. The plantations still exist. They're not as producing as much as they were before the hurricane, but they're still around. So I'll talk, as a pathologist, my main focus is diseases. That's what I enjoy the most. That's what I get paid to work on. So I'm going to talk a bit about the major diseases affecting cacao. It's estimated that about 40% of global harvest is lost to diseases. Of the diseases, these four here on the photo are going to be the ones that cause the biggest losses, particularly the one on the left, Phytophthora species. They're oomycetes. These cause disease on the pods as well as on the trunk. It's often referred to as black pod in English, and it's present. Phytophthora palmivera is the species that's present everywhere in the world. In all areas where cacao is commercially grown, that species exists. The other one is, to go back to black pod, in West Africa, when I had mentioned about the agroforestry systems in West Africa having an issue with disease, they have a relative, and it's called Phytophthora megakarya, and it's only present in West Africa. And it's a pretty big problem over there. It hasn't spread to other areas, but they sometimes specifically plant their trees further apart to dry, so it's not as humid, because humidity is what the pathogens like humidity, and they reproduce when it's very high humidity. And the other disease would be cacao swollen shoot virus. That one's also restricted to West Africa. It's not sure what the original host for that was. The latest research is thought that it evolved from viruses that were introduced along with cacao to West Africa hundreds of years ago. And the third one is Moniliophora rareri, that causes a disease called frosty pod. As you can see, the pod has the white frosted looking appearance. Those are spores. They're very resistant to desiccation and they live a long time. So it's easy to transfer that from your clothes to a new location or on your shoes. And that is only present in the Americas at the moment. And on the right is the pathogen Moniliophora perniciosa that causes witch's broom. So those two on the right are some of the main diseases that have limited cacao production in the Americas. And for the moment, they're still only limited to the Americas, although they are spreading throughout Central and South America to new countries. So black pod, in terms of yield losses, black pod, which are several Phytophthora species, these are the ones that cause the most estimated yield loss. The one in West Africa, there's several species that can coexist on a farm. Phytophthora megakarya and Phytophthora palmivera are the primary ones, and they can, under certain environmental conditions, they can outcompete each other, but generally Phytophthora megakarya outcompetes Phytophthora palmivera when they're present in the same location. It also has several other characteristics that make it more aggressive. It produces reproductive structures more quickly, and but it has a lower temperature threshold, so it can't survive very high temperature. So they have different characteristics that make them benefited by some of the practices. So in terms of the quantity of yield loss, it's about 800,000 metric tons per year. By far the most, in terms of global production, it's by far the reason for the biggest losses, even though if it coexists in areas like Colombia where it's present along with other diseases, usually frosty pod would be the one that causes the most loss. But this one is the most important just because it's present everywhere. And in addition to the pods that give it the name black pod, it also infects the stems. And you can get a range of symptoms. Like on the left, you get a gummy bleeding. Sometimes there's a salt like exudate with a sunken area. Sometimes you get, if you remove the bark, you can see the red parts of the stem. So there can be a range of symptoms you get with plant infection with Phytophthora. And here you can see the infection cycle. So as an oomycete, it has motile spores. So when it rains, this is how it's thought to spread. It's thought that with the rain, the sporangia or oospores are dispersed with rain or splashed from the soil, because they do also colonize the soil, and that splashes onto the trunk and infects the plants. About a decade ago, it was thought that it could only enter the trunk if there were wounds, but after doing research on that, we found that it can often infect plants, even if there's no wound. In the absence of wounds, it can enter and infect the trunk and cause canker. So some of the research that we've done on this is looking at what the inoculum reservoirs are, like where is the pathogen more likely to be found on a plantation. It's known that it can colonize the soil. It's known that Phytophthora in general can be in irrigation water. So we did some studies. This was done in Ecuador. Just using like soil and then filling in a pod to be able to, it's called baiting. So it's a way to isolate the organisms and see if they're present in the soil. And we use these methods to see just what the general proportion, where it was more frequently found. Surprisingly, it wasn't that common. I don't think we detected it in irrigation water at all, but it was pretty common in the soil. It was common in the soil, even in areas that didn't even on areas of the farms that didn't have disease, which was also surprising. So the cacao swollen shoot virus is the virus that's restricted to West Africa. It's in the Badnavirus genus. It has, as the name says, and on the leftmost slide, you can see the characteristic symptoms of this, the swelling of the stems. There are lots of viruses that affect cacao. This is the only one that causes stem swelling. So it's most notable for that, but a lot of the other symptoms it causes, there's overlap with other viruses. It's now accepted that this is actually a species complex. The most recent information I read was that it was about 12 species that caused this. 2 of them are considered very severe, where it will kill the tree within a few years of infection, but others are considered more comparable to the viruses that affect cacao in the Americas or in Asia. And it reduces, those other ones will reduce yield, but you generally don't see tree death. And the pod on the far left is circular, and that's one thing that starts happening when the trees are infected. The subsequent pods will start getting a circular shape. Obviously, there are some cacao clones that just naturally have pods like that, but it's something that happens over time with infected plants too. And these photos were taken on a trip to Ghana to look at some of the areas that were pretty hard hit by the cacao swollen shoot virus. On the right, you can see where the standing trees are dead. The government had gone in and they had an eradication campaign. Since a lot of the farms there are cultivated not by the landowners, but by people who rent the land, it's often, there's not the incentive to remove trees, because when they're infected, they will keep producing for several years, but at the same time, they're serving as a source of infection for other trees on the farm. So people who are just renting the land, they're hesitant to cut down the tree and then plant a new tree, and then they have to wait several years to get a harvest. So the government went in and was to incentivize them, would remove the trees for them and replant for them. But it had some limited success for a few years, but eventually it was about 2015, it became a big problem again. And so we went there to visit those. And one of the other diseases I talked about, so this is going to be one of the ones that's limited to the Americas. It's called frosty pod. You can see those masses of spores on the pod are white. It takes about 3 months to produce them. So the pod can be infected for about 2.5 months until you see any symptoms at all. And it's a range of symptoms from pod malformation, and eventually you'll get some necrosis, and then you'll get large amounts of spores produced. In areas where this has arrived, oftentimes the commercial industry will fall to practically zero. In this graph to the right, you can see when frosty pod arrived in Costa Rica, it was in 1978, and commercial production fell pretty sharply after that. This is one of the diseases that countries in the Caribbean, they spent a lot of time surveying for to try to detect it if it were to be introduced, since the spores are resistant to drying, they're resistant to light. It's easy to accidentally move this plant or to move this pathogen. Witch's broom, this is another pathogen that's also present in the Americas. It was first detected, I believe it was in Brazil. And it's not known what the original source of it was, possibly a host in the Amazon. But this one also takes about 3 months for infected plants to show symptoms. It'll start as a witch's broom, which means that a lot of small shoots develop on the stem. If you can see the photo that says 80 DPI, that means 80 days after infection, you'll start seeing those symptoms. And then about 3.5 months later, you'll start getting necrosis. The stems will die. In Bahia, in northeastern Brazil, a lot of farms were abandoned after this disease became established. Within the past 10 or 20 years, these farms have been restarted with plant material from the Brazilian government that they've bred for resistance to this disease. And this is another disease that's known. It's not very well studied because this one's particularly difficult to study. When it's called ceratocystis wilt, when the trees become infected, they die very quickly, sometimes within days. So whenever a farmer reports a tree like this, by the time you go out there to collect material for isolations, the pathogen usually isn't present anymore, and it gets taken over by other organisms. So this one's really difficult to study and to identify. But the map on the left shows you the distribution of it. It is present in, Haiti, the Dominican Republic would be the Caribbean countries that have that. It's believed to be associated with an ambrosia beetle, but it's not really known whether it's carried on the outside or on the inside. And it's just, it doesn't receive much study right now, but it's something that is considered a big problem, particularly in South America. So some of you might wonder why the US government invests into research on cacao. And even though there is commercial production here, the commercial production is not that large. Primarily, the reason for it is because the inputs that go into making chocolate, most of them are going to be sugar. You get milk, peanuts, and almonds, and other dried fruits. So those are products that American farmers produce. And so the chocolate companies are a market for American agricultural products. And so ensuring that there's enough cacao beans for the chocolate industry protects the market for US farmer for these other crops. So the approach for how to increase cacao production, it's projected that, so the demand for chocolate isn't keeping up with the modest increases in production. So this research looks at ways to increase global production of cacao. And the ways that this is being done is by developing germplasm that's resistant to these diseases. And the new germplasm is usually distributed free of charge to other countries because the goal is to have other countries producing a lot of cacao for the chocolate companies. And another way is to identify new and emerging pathogens that are limiting production that may not have been, people may not have been aware of before, and also improved understanding of these diseases. Oftentimes it's not, there wasn't much knowledge on what the transmission routes were for these organisms and what the environmental factors favoring transmission. So that's one of the ways that sought to increase production. So some of the activities for breeding, I'm sure many of you have had like those beautiful cherry tomatoes or beautiful strawberries that don't have much flavor, but they're beautiful and they're firm, but they don't have great flavor. So that's one of the concerns that people have with breeding for something like cacao or flavor. It's almost like wine. There's a lot of emphasis put on flavor profiles because they're trying to avoid having something where you produce a lot high yields, but without good flavor is what people are looking for. So there are whole research programs in Costa Rica, and Mars also has research programs where they focus on flavor and characteristics like that. So other things that they look at are cross compatibility. You want things that are ideally self-compatible, so you get more fruit set, but also highly productive and resistant to the diseases that I was mentioning. There are 2 genome sequences of cacao, and there are a lot of molecular tools that have become available in the past 15 years or so. And these are used to identify areas of the genome that are associated with disease resistance, which can then be used by breeders to more quickly screen populations for these characteristics. So this is some of the work that I was involved in, was looking at disease resistance. And oftentimes disease resistance is, you can go out to a field and evaluate it that way, but sometimes you don't know if the tree was actually exposed to the pathogen directly. So one of the ways that's considered the ideal to screen for disease resistance is to have artificial inoculations where you're physically putting the pathogen in contact with the plant. And so on the left, we did detached pod inoculations for Phytophthora palmivera, and that could be done in the lab. And for Moniliophora burreri, which causes the frosty pod, because it takes so long for disease to develop, that has to be done on the tree. But these studies, we'd go out to the field and specifically inoculate the trees with the pathogen. in Costa Rica. On the left are some of the detached pod inoculations. This was done in Costa Rica, in Miami, and in Puerto Rico. And it provided a lot of data on which plants in the breeding program had the best resistance to these diseases. And on the right are the field inoculations done in Costa Rica. So as I mentioned, Phytophthora also affects the stems, not just the pods. So to evaluate disease resistance of Phytophthora on stems, we do similar artificial inoculations where mycelia of the pathogen is directly put on a wounded stem. And then after a while, you come back, remove the bark, and check the expansion of the lesion. And that's your quantitative measure of resistance to Phytophthora canker. Once potential resistant plants are identified, the next step is to put them in field trials. So this was from a trip to Ghana where we went to visit some field trials of disease-resistant plants. So on the left, they're planted on a hillside. These plants I believe it was the first field trial, and they were reaching an age where they were going to be producing pods. So that's after doing the laboratory screening or the experimental screening, you actually put them out in the field to also make sure that they do well in these environments. So some of the other work that we looked at was looking at gene expression. So to be able to identify what are the genes involved that make certain plants resistant or susceptible, we use RNA-seq as an approach for that. And it also looking for molecular markers associated with resistance. So one of the things that I worked on, this was about 5 years ago. I did disease surveys in a lot of different locations. So most of the farmers, they use knowledge just that's been repeated about what diseases are present in an area, but there's a lot of overlap for symptoms. So certain symptoms can indicate more than one plant. And there was a lot of conflicting information about what diseases were really present in each area. So I did disease surveys in several countries, in South America, and Puerto Rico, and in Hawaii. I would go to the farms, take samples of symptomatic material, do isolations, and I had a pathogen import permit. I would bring the pathogens back to the US to identify them based on genetic sequences. So for symptomatic stems or soil near disease plants, that has a baiting step, and then you wait for lesions to develop before plating it on media. So that's the isolation process for stem pathogens. And for pathogens directly from pods, usually can just do the last step, which is plating them directly on the media after surface sterilizing them. So in Hawaii, one of the things we found that was surprising was that a lot of what was being referred to as black pod actually wasn't caused by Phytophthora species at all. We did find Phytophthora palmivera, but it was pretty rare. In my hypothesis is that it was rare because the farms were just colder overall. They had lower temperatures, but we found Fusicoccum parvum, which is a fungus not known to be a pathogen to cacao before this, and the symptoms were indistinguishable from Phytophthora palmivera. That would be something that you wouldn't be able to identify without really doing isolations. And in Puerto Rico, most of the things that would be referred to as black pod were caused by different funguses with diaporth species. More than half of the necrotic pods, the pathogen turned out to be diaporth species. And in this disease survey, I also found a cacao mild mosaic virus on a commercial farm. This was the first. Before this, it was thought to not exist outside of Trinidad, and it was thought to be mostly eradicated. The government in Trinidad had a pretty large eradication campaign, and it hadn't been detected outside of there. So this was a surprise. Yeah, so this was in the commercial farm and also in the USA germplasm collection. Those would be the symptoms on the right, where you have a modeling mosaic of the pod as well as some chlorotic modeling on the leaves. In some of the entomological research, for insects, at least in the Americas, beetles, they cause a lot of damage on stems and also pods, and they're thought to be associated with disease incidents in the field because they're wounding the surface of the pod, which makes it more likely for organisms to get in. And Lepidoptera are a big problem. In Asia, there's the cocoa pod borer. In South America, there are a couple different genera of Lepidoptera that cause similar damage, slightly less damage than the cocoa pod borer. But on the far right, you can see that would be a Lepidoptera that got bored into the pod and damaged a lot of the inside. So this work looked at going out and collecting insects, finding ways to identify them that didn't require taxonomy, because collecting insects in the field and then shipping them to another country, they were always shipped dead already. They often get damaged in the process. So not needing to look at very small structures was beneficial. So we developed protocols for molecular detection of those. And for new and emerging pathogens on cacao, because these disease surveys looked at everything, we ended up finding some interesting things. So obviously the Phytophthora canker was more of a problem that had been expected. These intensive full sun plantations in Brazil and Ecuador were planted with a clone called CCN51 that was known to have resistance to other important diseases. And it was a surprise because it was being reported to have really high incidence of Phytophthora canker. So in conjunction with Mars, I went to Brazil and Ecuador for a research project looking at what some of the causes were and to identify what the organism causing this was. And so one surprise, we did find a new species of Phytophthora. It ended up being called Phytophthora theobroma. This one was present in Brazil. I believe it's since then been found in Colombia on cacao. And it tolerates a wider range of temperature. And depending on what the clone is, it can be sometimes more aggressive, sometimes less aggressive. So that was surprise. And we did also do some screening to see if CCN51, which was being chosen for disease resistance, was possibly more susceptible to canker than other clones. And that work is still ongoing. And one of the other emerging diseases was cacao mild mosaic virus, the one that was detected on the commercial farm in Puerto Rico. That was in 2021. So the primary symptoms of that one are going to be mosaic on the pod. There's also mosaic on the leaf vein banding. If the tree is pretty heavily infected, you'll get like the small pods in the middle. They'll be small and misshapen. You can see the mealybug evidence on the surface of those pods with the white. Those are egg masses, and those are ripe pods that just stayed very small. If you plant the seeds from them, the seedlings that grow from that are infected with the virus as well. Because there's a lot of things that can cause pod symptom or leaf symptoms on cacao, usually the indicator or the advice I give to people that are wondering if they have it is to look for pod symptoms. So there aren't that many things. There are lots of things that can cause coloration changes on leaves, but there are very few things that will cause coloration changes on pods. So if you see mosaic on pods or chlorotic islands or any unusual coloration, it's a good indicator that you have a virus. So people that would send me samples to do genetic studies on just the genetic diversity on the virus, they would often send me samples from their pods themselves, pods that had these kinds of symptoms. So the history of cacao mild mosaic virus, like I mentioned before, it was discovered in Trinidad, and then there was an eradication campaign. Then it was found in the International Quarantine Center in the UK, which is an intermediate quarantine center. And after it was found there, the material was destroyed. And then in 2019, it was found on the commercial farm in Puerto Rico. Then it was found in the USDA ARS quarantine greenhouse in Miami. And we were having some trouble with a previously existing molecular test. So I did develop a molecular test based on several strains of the virus that I had sequenced. And that test has since been used to detect the virus in Indonesia, where it's found that it's pretty widespread. It's also been found in Hawaii. This was the development of the virus. It's been used in several countries, and these are the sequences that I based the primer design on. So the cacao germplasm collections, one of the ways for increasing global production of cacao is to have improved germplasm planted. And so the germplasm exchange happens from gene banks or from germplasm collections. It's sent through intermediate quarantine centers before going somewhere else. And since the discovery of CAMMV in Puerto Rico, the collections have been tested, and it was found in all 3 of the USDA ARS germplasm collections. And now the major collections in Costa Rica are being screened for this as well. One of the things we were concerned about, since at least in the Americas and I believe most of Asia, the plants that are used for farms are grafted plants. And the rootstock will be grown from seed, and then the top will be a cutting from whatever desired clone before planting. But even in areas where viruses were known in cacao, rootstock was never screened for viruses. So I did a transmission study to see if it was what the rate of seed transmission was. It varied quite a bit, anywhere from 17% to over 50%. Depending on the location. These were field trials using populations in different geographic locations. And this is also some work done on the insect vector. So it's the cacao malmosaic virus is transmitted by mealybugs, several different species. And all we had to go on was publications out of Trinidad that were I believe they were from the 1960s. And since it had been mostly eradicated, not much work had been done on that. So since we had a pretty heavy outbreak in a greenhouse in Miami, Florida, we collected insects and just identified them to species based on genetic sequences to find what species are associated with this virus in that area, which would be like the Caribbean and Florida. There were some new, so we didn't do transmission tests because that wasn't allowed under our permit, but hopefully someone else can do transmission tests in a place where these insects are already present. And another study looking at the epidemiological study was looking at what are other hosts. So in places like, for example, Puerto Rico, there are lots of plants in the Malvaceae family that are planted ornamentally or just present in the forest environment. And so we wanted to see what other plants could also be infected with cacao malmosaic virus. So these plants were collected just naturally growing. We didn't artificially inoculate them. This table shows in which ones we did find the virus. There's a very small number of each plant collected, but there were several species of plant that were found to be infected with the virus and could potentially be pathogen or inoculum reservoirs for cacao farms. And so for many of you, I assume that either you're associated with botanical gardens or you're probably into gardening or having plant collections. So I put this together. This will be something where if you have cacao trees or you're trying to grow them at home or in a greenhouse, these are going to be some of the issues that would be pretty common. So on some of them, this is often people send me photos of these, asking me if it's a black pod. So if you see there's some necrosis on the pods, but this is what we call Sherrill wilt. It's believed to not be pathogen related. There are a lot of theories, but it's thought that the tree produces more pods than it can sustain or bring through the full physiological stages. And so it begins aborting them. And as it aborts them, they start decomposing. So that's Sherrill wilt. And I would say it's common on plants grown in either botanical or just under artificial conditions where they could be related to light level. There's a lot of theories. None of them are perfect, but that's a common problem. Another one that's seen pretty much everywhere, even in greenhouses, would be branch dieback and pod necrosis. It's usually caused by numerous fungi, and particularly Botryosphaeridae family. The lesions look very similar to Phytophthora. It won't kill the tree. It helps to prune back diseased parts. Definitely remove pods if you get them, if you get any necrosis on them. And so these are going to be some examples of what you could see in some non-commercial plantings. On the left is Phytophthora palmivera. On the right is Lasiodiplodia. I only know this because I isolated the organism and sequenced it. They look very similar. Both of them, if you remove the pods, you get rid of the inoculum for the most part, or at least reduce the amount of inoculum. These are other photos that I've taken where people thought they had black pod or another disease. So on the left, this is just what we call an overripe pod. It wasn't removed from the tree, it began decomposing. So it's not a pathogen, but you will get fungi. You will find fungi in there if you try to do isolations. And the one on the center was wild boar, or just any kind of person walking by the tree, or it's physical damage on the surface of the pod. And on the right are pods that died for some reason. Can't really tell, but they just dried out on the tree. Sometimes that happens with overripe pods. Probably not a disease, but it's impossible to say at that point because they're dry. And so I talked about a lot of different work I did, and there were a lot of people involved in that. Involves researchers in many different countries, people at Mars with the private company, and VMR Consulting in Puerto Rico. And so, yeah, a lot of people, a lot of ongoing research, a lot of insects and a lot of pathogens. So if you guys have any questions, I tried to save some time. Let's see.
Alina, thank you so much. That was fabulous. Yeah, boy, I was amazed. I always think about chocolate, but I never thought like, so why is the US so interested when production is so low? The idea that it has to do with things like sugar and almond production and things like that. That was really fun to hear. So yeah, I think we'll just open it up here to questions. And people can either raise their hand, they can put questions in the chat, or just unmute.
Alina, I have a question. So I'm a little bit confused about how the disease might be prevented from moving out of roots into the shoots, or I mean, you know, how it seems like depending on what it is, it may either be able to be reinfecting from a rootstock or not. So this last mosaic virus, you were showing that the roots would could reinfect the top graft, right?
So with the cacao mosaic virus, if the rootstock, the one that you use as the base, is infected, when the plant material fuses, the virus moves into the upper one. If you have an infected rootstock, there's no known treatment. The way to prevent that is to screen your rootstock before doing the grafting, which was something that wasn't done before because it was thought to not be seed transmitted. So yeah, so now one of the easy ways, or the fast way to do it, is the mother tree that you're getting the pods from to plant for rootstock. You screen that, but ideally you would screen every single plant that you're going to use as a rootstock, because nobody's tested whether it's pollen transmitted. So we know it's transmitted if the mother tree is infected, but it's possible that the mother tree isn't infected, but if it receives pollen from an infected tree, that could be a transmission route. It just hasn't been tested.
Thank you.
Alina, I've always, when I was at the Botanic Garden, I always considered myself a plant storyteller. And I hoped that I was accurate. And so I've always been fascinated with the pollinator, the midge, and the trait of the cauliflory, where the flowers grow on the trunk. Is there a direct relation because they are wingless that the pollinator can just move up the trunk, and so that there's some evolutionary between that trait and the pollinator?
I'm not sure. What had always been explained to me was that, if I can go back, was he's the entomologist. He thought that possibly the midge was a pollinator, because to get under that, the covering of the petal that's directly over the anther. So if you do the artificial pollination, you have to specifically pull it back. So they were thinking that possibly some of the winged ones couldn't fit under there. But I don't remember anyone talking about that specifically cauliflory, a possible association between that. But they did think that it was just a lot of the insects couldn't get in there, which is where the pollen was. Did you ever do artificial inoculation at the Botanic Garden?
There was, I don't know about that. There was a lot of hand pollination. And yeah, I mean, that's essentially how we got our pods. But I don't know about inoculation.
Oh yeah, I meant artificial pollination. Yeah, not inoculation.
Oh yeah, all the time. That's essentially, I mean, every once in a while, you know, something would pollinate it, but for the most part, we would get it through. Yeah, Sean, our arborist in the early morning would go down to our other tree and bring over the flowers and perform that nice little, you know, plant sex, flower sex.
Yeah. Yeah, even after pollinating them, sometimes they'll take and then they'll still abort. But yeah, I have had some trees that, even though they're supposedly not self-compatible, I have had some plants in greenhouses that were isolated from the rest, and there's definitely no insects in there, and they've developed pods. Not very many, like one or two. So there must be some variation in either what can pollinate it or just whether they're compatible. Well, there are some that are self-compatible. It's just considered pretty rare.
Are there any efforts to do disease removal through isolating buds the way they do for citrus at the Riverside location?
Through doing what? Could you repeat that?
So for citrus, you know, citrus has a lot of diseases. ARS has a program at the Riverside collection to do basically bud culturing to try to get rid of a lot of the viruses and other pathogens because that's also a clonal crop.
Yeah, so I know with the cacao swollen shoot virus, there was some work being done. I think it was in Ghana, and they were doing tissue culture from the bud to try to get rid of the virus. And the success rate, I think they had about close to 80% success rate with clearing the virus from those plants. I think USDA ARS has a small trial that they're doing in Puerto Rico to try to get plants without cacao mild mosaic virus, because some of the plants, when we found it in the quarantine greenhouse in Miami, some of the plants, they're pretty valuable. So yeah, I suggested before discarding them, maybe you can try doing tissue culture. When they're that valuable and they've spent that many years in quarantine, even though tissue culture, I mean, it's pretty labor intensive. It's something that I think could be a possible option.
Alina, I have another question. You found these new species in Hawaii and Puerto Rico. Do you think the current kinds of treatments and sanitation measures are going to work for them, or should we be really worrying about that?
You mean for the fungi?
Yes.
Yeah, so for those, they were, those were species that had been described, I believe primarily from, the Neophysococcum was primarily from South Africa. They seem to respond to just the common fungicides. One of the things that we did find notable was that, at least in South America, a lot of fungicides, instead of applying a general fungicide, a specific fungicide for oomycetes, because they assumed it was Phytophthora. They were applying chemicals for oomycetes. The commercial name is Aliette. It's, I believe, aluminum phosphatase, something like that, and which is not, it's not effective for fungi. So that's one of the reasons I do think it's important to specifically go to the locations and find out what it might look like black pod, which is normally caused or thought to be caused by Phytophthora, but it may actually be caused by a fungus. And if you do have fungal sources of pod necrosis, the aliette wouldn't be an effective measure. So it would just be a matter of where if you know what pathogens you have, you can pick something that's effective against both.
Yeah, that makes sense. Thanks.
After the eradication, how long does one have to wait for growing? Could you grow trees again in that same area? And if so, how long would you have to wait?
So the eradication program in Trinidad, I'm not sure if it wasn't well documented because it was so, I want to say it was in the 60s, 1960s. So I'm not sure how they waited. I know they went through the islands and they removed any tree with symptoms, but newly infected trees, they normally don't have symptoms for a while. So it's easy to miss. But in Ghana, where their eradication campaigns they're more thorough and they're well documented. I believe they did not change. There was no waiting period. What they did was remove the symptomatic tree. They removed trees within a buffer around it. I forget the distance. And then the government would provide new seedlings and for the farmer to plant, and they would be planted right away. So there was no waiting period because the tree takes years to get production from. So they didn't want to take away their livelihood. So they would provide and plant new trees with no waiting period.
Question for you, Steve Lonker here. Has there been any research in making use of the plant's microbiome for pest management strategies against Phytophthora?
Yeah, actually, I cut the slides out because it was getting unruly, my
Really?
presentation. But yeah, so I was going to talk about some work that we've done on looking at microbiome, not necessarily as a treatment, but one of the things I mentioned, so there was a scientist at Mars who's an expert in microbiome. And one thing I mentioned to her was that I noticed when I would go and I would do these inoculations, like artificial inoculations, and it could be the same pod, the exact same clone of a tree. And some pods would get huge lesions, but other times from the exact same tree even, it wouldn't get a lesion at all. And I just wondered, like, how can there be so much variation when we know like their clones, like they're genetically identical? And I was like, could it be, either something in the environment that influenced the microbiome or something like something in more detail, because there's a huge variation. And so we did a study looking at two different trees. They were growing really close to each other. So it was the same environmental conditions. One of them, the pods from that one developed significantly larger lesions than the pods from the other tree, which was genetically identical. And so we did find some differences in, I don't have the summary with me, but yeah, we found some differences in the microbial communities in terms of using that as a way to manipulate it to then prevent disease would be a little further off. But yeah, there is, her name's Jennifer Schmidt. She works with Mars. So she was doing some work on that, looking at the interaction of that with disease incidence in the field, as well as productivity. So yeah, it's an interesting area.
Yeah, cool.
Hi, Ed Barrows here. I'm wondering if people are concerned about global warming and cacao production right now.
Yeah, in terms of the effect of cacao on global warming, or the effect of global warming on cacao?
Well, the effect on cacao diseases in particular.
Yeah, so that's that would be something that would change so when the two phytophthora that I was talking about being present in West Africa, specifically in Ghana, where so the changing environment, changing temperature will affect their interaction. In that case, I'm not sure obviously it'll stress the trees, which always makes them more susceptible to disease, but in that case, there will probably be some interaction. It might reduce some of the issues they have with oomycetes because it'll be higher temperature, and the more aggressive oomycete has lower temperature tolerance. But I imagine it would, if the plant is stressed, there will be more issues with the fungal dieback. This would be my theory. Probably also if the plants are stressed, they'd be more affected by insects, especially ambrosia beetles that are drawn to stress related volatiles. So yeah, that would definitely affect it.
Okay, thank you.
Maybe time for another question or two. Anybody?
So we are ready, and of course, I'll have a conversation with the company to write the line, from London. And I'm going into a cabinet room right now to talk with my colleagues about the cabinet evacuation.
Council is publicly saying that there's nothing that people can do to avoid the tariff. This is not a negotiating. In reality,
Sandra, do you have a question? Or
we're able to negotiate a deal the same day tariffs were supposed to go in effect, or basically a deal. And similarly, this time we have Trump making a similar statement saying this is not a negotiating tool. There's nothing they can do.
Maybe she needs to be muted.
Sandra, I can't tell if you have a question or you're just unmuted.
Well, I think this is the biggest scientific discovery of my lifetime. And it's
I think unmuted.
with the great scientific discovery of all time.
Okay.
Discovered by the discovery of
Well, if there are no other questions, Elena, I just wanted to say thank you so very much for joining us this evening. We're thrilled that that you could be with us online, because if we weren't online, you weren't going to be with us. And so yeah, we thank you for staying late at work and joining us and bringing us this great information. So yeah, thank you very much.
Yeah, thank you for the invitation, and thanks for listening. I love talking about research, so yeah.
And save our chocolate. Save our chocolate.
Very, very important.
I've got a personal stake in this one.
Yeah. Same here. Yeah. Okay, well, have a good night then. Thanks.
Yes, thank you so much.
Bye.
Bye-bye now.